So DNA is a double-stranded helix in the nucleus of our cell. The DNA and RNA don't really do much on their own that we know of right now in the human body. They need to get translated into proteins, and the proteins are the stuff that actually do stuff. Like proteins are enzymes and cytokines and all those things. Those are proteins, right? So a double-stranded helix of DNA gets translated into a single strand of RNA, and then that single strand of RNA gets translated into proteins and peptides, right? So it's like translating English to Spanish to Chinese. If any malfunction happens in any of those translation processes, the end product is going to be faulty and it needs to be recycled, right? It'll be gobbled up and the whole process will go over again. The problem with screwing around with RNA and DNA technology in humans are many. It's absolutely insane to be messing around with this type of RNA and DNA technology right now. Depending on the research that you read, 90 to 95% of our DNA, we have absolutely zero idea what it does, meaning that 90 to 95% of our RNA, we have zero idea what it is. If you guys have heard of the junk DNA that we all learned about in school, what we didn't learn about in school was that's 95% of our DNA. We do not understand what 95% of our DNA and RNA do in our body. We know that if you remove it, you die, so it's obviously important, but we have no idea what it does. So to be messing around with these processes in the body is absolutely insane. On top of that, the epigenome, which we'll get into in 1 second, and it's much more discussed in the epigenome class, the epigenome can be inherited. So these are changes that you can make, markers, tags that you can add to your DNA that can actually be passed on to one generation from the other. So if you have your RNA and DNA screwed with as an adult, that is likely to be somehow passed on to your offspring, right? It's very, very dangerous stuff. Not good. So here's, again, just another relatively easy way to look at how DNA gets read into proteins, right? So we have our chromosomes in the nucleus of our cell. We have mitochondrial DNA from the mom, nuclear DNA from the dad, right? So the double-stranded helix gets translated into a single strand. So double-stranded DNA gets translated into a single strand of RNA. That RNA gets translated into a protein. The proteins are actually things that do stuff, right? So they're made up of different codes. You get different letters for the DNA and the RNA. Again, all of this stuff will be gone over in depth in the epigenome class. But this is why, you guys, this is why eating well and supplementing well works, and it's also why eating poorly and not supplementing is bad for your health. The reason that eating good food does good things for us is because it changes the epigenome, which is a level above our DNA, our genome. So the epigenome controls how our genes are read and expressed, right? So if you're eating food that your body doesn't like, it's going to alter the epigenome and it's going to change the way that our genes are read and expressed, or if they're expressed at all, right? So there are certain gene anomalies that many people have. They will get expressed if you eat certain processed foods, and they won't get expressed if you don't. So that's the difference between health and sickness, right? So here is a good example of protein folding, right? The left picture is just kind of the primary, secondary, tertiary, and quaternary structures of protein. The right picture here, you guys, this is the process of what happens when our DNA to RNA to protein transcription and translation goes awry, right? So what protein a certain gene is going to produce totally depends on the environment, the cellular environment that the cell is in, right? So each gene can produce lots and lots of different proteins. It only produces the proper protein if the cells are in the proper homeostatic environment. If our cells are too acidic or too basic, or we have chemical concentration abnormalities, The proteins that are produced by the DNA to RNA to protein transcription and translation process are going to be faulty, misfolded proteins, like the alpha-synuclein protein that we see in the brain with neurodegenerative disorders, right? Or the beta amyloid plaques. Those are misfolded proteins that our body has not been able to gobble up. So if you have enough ATPs and your mitochondrial health is sufficient enough, when our body makes misfolded proteins, it recognizes it, it gobbles up the proteins, it throws them out, and it starts the process over again. The problem that people have with things like Parkinson's and Alzheimer's and COVID and all this other stuff is that if our body is in a state of energy deficiency, which again, 80+% of the country are in a state of chronic energy deficiency, our body only has a finite amount of energy to use. If we run out of ATPs, then our body and our brain has to start making hard subconscious decisions of where to send that energy. But the bottom line is there's not enough energy to properly recycle or clean up the garbage, more or less, in the cells. So as we become energy deficient, our cells are in a worse environment. They start to misfold proteins more. So the translation or the translation from DNA to RNA to protein malfunctions more. In a healthy state, it would malfunction less, and we would have the energy to gobble up and throw out the misfolded proteins. As we become energy deficient, we have more misreadings, we have more misfolded proteins, and we run out of energy to recycle those misfolded proteins, so they start conglomerating in nerves and in nervous tissue, and that's what leads to things like the beta amyloid and alpha-synuclein plaques that we can see in Parkinson's and Alzheimer's disease. So, one really, really cool study, and this is just a good example of how little we actually know about the human body. About 5 years ago, a group in France found the alpha-synuclein protein that's related to neurodegeneration that we typically see in the brain. They found that protein already misfolded in the intestines, right? This has never been seen before. So what these researchers did is they radioactive biomarked the misfolded proteins. Those misfolded proteins traveled up the vagus nerve and ended up in the brain. So now we know that there's definitely numerous types of Alzheimer's and Parkinson's. There's some that start in the gut, there's some that start in the brain, and I'm sure there's other pathways that those misfolded proteins end up in the brain, but they don't all start in the brain, right? So this is just another good example of how complicated our body is, and to be messing around with our nuclear material like DNA and RNA, it is impossible to say that that is safe for anybody, because again, we don't know. The government says this on their website. We have no idea what 90 to 95% of the DNA is responsible for. Therefore, we have no idea what 90 to 95% of the RNA is responsible for. We have no idea what we're doing with it. If our epigenome is malfunctioning or not in the correct state, our DNA cannot be read into RNA, and that can't be read into proteins properly. So basically, our body is not going to produce the stuff that it needs to function well, and it's going to produce stuff that leads to more dysfunction. Right? So this is really, really cool, you guys. This stuff drives me nuts because this should be all widespread knowledge in the medical community and in the general population, but it also gives me a lot of hope because we have the ability to treat people so much better than we are right now. We have the ability to make people so much healthier than we are right now. All we need is the information and knowledge and understanding to put it into practice. Right? So this is what I'm trying to do with these classes is give people a condensed version of all the important information that you need to treat your patients better and to treat yourself better, right? So there's a lot of confusing stuff in a lot of these research papers. Again, I've tried to put it into the most important, understandable English that people can comprehend and put into practice. This is just a very brief introduction on the epigenome. So what is the epigenome and what does it do, right? So humans have trillions of cells in our body. All of our cells, as far as we know, have the same genetic material inside the cell. And this is the crazy cool thing about the human body. It's just so incredibly complicated. In each cell, we have about 6 feet of DNA that's wrapped around something called a nucleosome. And then for the genes to be read, the DNA that's wrapped, just think about like a bobbin and thread wrapped around it. if we need certain genes and proteins to be expressed and produced in our body, our nucleosomes and our histones unravel that DNA so the cell can access a little piece of the DNA to read, and then it gets rewound up, and then it can get unwound and a different section can get read. It's incredibly complicated. It's so cool. So all of our cells have the same genetic material in them, but different cells produce different things and they turn into different cells, right? So like our pluripotent stem cells are endoderm, ectoderm, and mesoderm, the three types of primary cells that we have when we're first conceived inside our mom's belly, when the sperm meets the egg, right? Those are our pluripotent stem cells, and those can differentiate into other types of cells. So liver cells and kidney cells have exactly the same genetic material inside of them. It's just different parts of that genetic material are being expressed, which makes them into different types of cells with different functions. It's so incredibly amazing, right? So this was first kind of thought about / discovered like in the 1800s, right? When people like Mendel and Darwin and all those people started looking at inheritance, there used to be a big issue called the missing heritability problem, where we used to think that whatever the DNA said is how you were going to be, or what proteins are going to produce. And we know that to be patently false now. So the healthier we are and the better things we put into our body, the more accurately our DNA gets read into RNA and then into protein, the less cleanup our cells have to do, the less energy it requires, and the more energy our cells and our brain has to function normally, right? So chronic inflammation is a really good example of this. Again, one of the hallmarks of aging. Chronic inflammation is not a normal state for human beings, although the vast majority of the country is living with chronic inflammation. So people with chronic inflammation, and this is talked about in dry needling literature, manipulation literature, nutrition and supplementation literature, there's a handful of primary pro-inflammatory proteins, cytokines, other stuff that cause excessive inflammation, right? So interleukin 1, interleukin 6, tumor necrosis factor alpha, C-reactive protein. These are all commonly elevated proteins that cause inflammation in the body that we see in mental health disorders, in physical health disorders. Basically, if you're a regular human being and you're not eating well and you're not exercising and you're not taking care of yourself and you're actually living life, so you have stress, you have environmental toxins, you have other factors that are stressing your body and stressing your brain, this tends to lead to a chronic overexpression of the interleukin genes that cause the pro-inflammatory interleukin proteins, right? The cytokines, interleukin 1, interleukin 6. Those are the pro-inflammatory interleukins. There's also anti-inflammatory interleukins like interleukin 10 and interleukin 17 and some of the other ones, right? So again, by eating bad food and by not being in good physical condition, we are causing chronic inflammation in our body by expressing genes too often that should not be expressed. They're just being expressed because the cells are in a state of stress and dyshomeostasis or non-homeostatic condition, right? So this is modifiable, and this is why the epigenome is so cool. The epigenome, this is a good picture here, right? So you guys can read the little left, that's just the blurb from the picture, but this is a good visual representation of the epigenome. So we have chromosomes inside of our cells, right? The ends of the chromosomes are the telomeres, or like the little caps on the ends of the chromosomes, right? So telomeres, in general, the longer our telomeres are, the more replications a cell can have, the longer the cell life, and the more ability it has to improve health. So the longer our telomeres, the better health span and lifespan of our cells, right? So almost all cells, most cells have something called a Hayflick limit, or the maximum number of times a cell can replicate. Once the telomere gets gobbled up, the cell can't replicate anymore, it loses energy, and it goes into autophagy, where our body gobbles it up, right? However, if we're eating bad stuff all the time and not exercising and not doing the proper things for us, the epigenome gets modified, which modifies the way that our DNA gets read, right? So the epigenome controls how our DNA gets read. So again, there's things called histones and nucleosomes. We'll get more into this in the epigenome class. But based off of what we're putting into our body and how we're acting, we get little tags put on. So if you think about the double helix, just think about like little marbles getting glued onto the outside of the double helix. Those are epigenetic markers. Depending on the little marbles that get glued to the outside of the double helix will determine how the DNA gets unraveled and read and how it gets expressed, or if it gets expressed or not. So this is why it's so cool, and this is why nutrition and supplementation work, you guys. It's because they modify the epigenome, which modifies the way that our DNA gets read and how accurate our DNA gets translated into RNA and protein. And it certainly has an effect on the 90 to 95% of our DNA that we have absolutely no idea what it is, the so-called junk DNA, which is obviously not junk. Again, if you take it out, you die, so that's not junk. We just don't know how it works, right? So in general, that's how the epigenome works. We'll get into this in detail in the epigenetics and genome class. Here's another picture, same type of picture. So there's the chromosome, right? You get chromatin that comes off the chromosome. There's methyl groups and acetyl groups and all this stuff. This is a little bit better picture of nucleosomes and histones, right? So the little histones are the little circles, that whole thing. When you get 8 histones together, it can make a nucleosome. That's what our DNA gets wrapped around, the nucleosomes and the histones, and then it unravels to read certain things and re-ravels. It's very, very amazing process. It's incredible that all this stuff happens. But again, what's really cool about the epigenome is it's modifiable. So this is why it's so important to get good epigenetic tests and blood testing done and genomic tested, so we can see, do we have as an individual some type of genetic anomaly that's going to affect the way that our DNA gets read? And if we do have that anomaly, there are plenty of ways to go about making sure or trying to ensure that certain things or certain genes don't get expressed or overexpressed. A great example of this is folic acid. Right, so we mentioned this a little bit before. Again, this is covered in depth in our nutrition and supplementation class and the epigenome class. So just real briefly, methylation is an epigenetic process. You put methyl groups onto the DNA, which changes the structure of certain molecules and things like that, right? So if we want to have a properly functioning epigenome as far as methylation goes, and there's a bunch of other processes, we must have sufficient levels of a couple of things in our body. 5-MTHF, 5-methyl tetrahydrofolate, is the bioactive form of folate, right? So folic acid is not usable in the human body. You must have a certain type of gene in your body to be able to methylate or alter or convert folic acid into the usable form, which is 5-MTHF, 5-methyl tetrahydrofolate. 30 to 60% of the United States has a gene anomaly that makes it basically impossible for our body to efficiently convert folic acid into 5-MTHF. Right? It's called the MTHFR gene mutation, or methyl tetrahydrofolate reductase gene mutation. It's an enzymatic mutation that does not allow people to convert folic acid into folate. It's really incredible that humans can convert folic acid at all, because it's an alien substance not found anywhere on the planet. And there's only a couple countries in the world that allow folic acid to be put into the food, because most people have taken it under consideration that over 50% of the population guaranteed is going to have a problem with eating folic acid. And folic acid is put in every single non-organic grain that we have in the United States. So again, this is testable. You can find out if you have one of these gene mutations. I do for sure, right? So just real quick example for me, if I eat stuff with folic acid in it, the back of my head and my ears get itchy within like an hour, and they'll start to flake, and it's all nasty, right? If I don't eat folic acid, that doesn't happen. The reason is because I do have one of the MTHFR gene mutations, and my body can't efficiently or can't at all break down folic acid into methylfolate, which is the usable form. Right? So it's just like putting water, it's like putting a few drops of water in your gas tank every time you fill up your gas, right? The car's made to run on gas. It's not made to run on folic acid, or not made to run on water. Our body is made to run on 5-MTHF, 5-methyl tetrahydrofolate. It's not made to run on folic acid. So it requires extra energy of our body. It requires extra energy of every human's body to be eating folic acid, because it has to be converted through multiple steps into 5-methyl tetrahydrofolate. If you're just eating methyl tetrahydrofolate itself, 5-MTHF itself, your body does not have to break it down into anything. You're putting the bioactive usable form into your body already, right? So this is one of the things that will lead to elevated homocysteine levels in your body, amino acid dysfunction, cardiovascular disease, mental health disorders, all types of stuff, right? Again, this is something that 30 to 60% of the United States suffers with, and I guarantee you 0% of patients in the United States have ever been told this by their doctor, because I've never heard this from any doctor, right? It's very, very disturbing, except for people like Mark Hyman and Dr. Peter Tia and stuff like that, right? Regular primary care doctors or mainstream medicine don't know this stuff exists, which is absolutely unacceptable. Okay, so to have a properly functioning conversion system from folic acid to 5-MTHF, or for a bunch of other things that need to be methylated, we need to have proper amounts of active folate, active vitamin B12, active vitamin B6, active vitamin B2, active magnesium, betaine, and vitamin D, right? So again, 70% of the United States population is deficient in magnesium. 95% of the population is deficient in vitamin D. So just based off of those two facts, if you are eating folic acid and you actually do have the ability to break down folic acid into 5-methyl tetrahydrofolate, there's a 95% chance that your body can't do it properly because you're deficient in vitamin D. So being deficient in vitamin D does not allow normal methylation of proteins and anything like that in our body, normal methylation of RNA, normal methylation of DNA, if you're vitamin D deficient, which 95% of the United States population is, right? Our methylation process is not functioning well, meaning we're getting way too many misfolded proteins that aren't doing what they're supposed to do. Our body has to gobble it up and clean it all up. That all requires energy. It drains our autonomic nervous system reserve well of ATP energy. It drains that, and at a certain point, the well runs dry, and then our body has to make really hard decisions on where to use the available energy, because there's not enough energy available for everything, right? So that's a very, very simple, quick rundown of one of the processes in the epigenome that is controlled by the amount of proper nutrients that we do or do not have in our body, right? So again, this is why eating well makes you healthier, and it's why eating poorly makes you sicker. If you can get your patients to understand the why behind it, they're much, much more likely to follow instructions and do good things for their own health, right? If they don't, at least for me, if I don't understand why somebody's telling me something, I just basically don't listen to it, right? So I like to know why, and I think this is just a regular human condition. Unmetabolized folic acid syndrome. So this is something that happens, like I mentioned before, with gluten protein and the lactose protein, right? If you're eating a ton of folic acid and your body can't break it down, you actually, and it's a little counterintuitive to think about, you actually end up with excess 5-methyl tetrahydrofolate levels in your body. So I know I just said that that's the bioactive form and folic acid is the non-bioactive form. But what happens, like so many other synthetic molecules that we're not used to eating, they have a higher affinity for the receptors on the postsynaptic cleft side than the natural substance does. So if you're eating a lot of folic acid and you have one of the gene mutations, you actually do have methyl tetrahydrofolate in your body, but our cells have a higher affinity for folic acid. So then you end up with all of our postsynaptic clefts being being utilized by folic acid, which is not an actual usable form, so they're just taking up space and energy. And then we get excesses of methyl tetrahydrofolate and homocysteine and some other proteins and peptides, which just cause widespread dysfunction in our body. It leads to decreased ATP production, hypoxia, mental health disorders, all sorts of stuff, right? So again, this is fixable stuff for everybody in the United States if you have the proper knowledge and understanding of what you need to get tested for and how you can fix it. And again, this is not information that anybody has. I mean, more or less, 0 doctors in the United States know anything about any of this stuff. There's some of them, obviously, but there's very, very, very few. Right? And basically 0 of the general public knows about this. So this is why it's so exciting for me, you guys, is that we have the ability to improve our patients' health and to improve our own health so much more than people think we do. It's just basically not knowing what you don't know, or knowing what you don't know. That's the problem. All right, you guys, so that's a brief rundown of epigenetics, nutrition, supplementation, vitamins. We didn't really talk about peptide therapy. That will be covered in its own class, right? So this is a brief introduction. It's 5 CEU class. If this was interesting to you guys, and it seems like something that you would want to implement into your practice, which I recommend that everybody does, if you want to make your patients healthier, or if you're not a medical practitioner and you want to be healthier, this is absolutely 100% vital information to have, because without it, you just can't rely on a regular mainstream medical doctor to know any of this stuff, because they don't, right? So if you just want to hope that somebody's treating you well, don't pay attention to any of this stuff. If you want to take your health and the health of your patients into your own hands, this stuff is 100% necessary to know, right? So in our next classes, you guys, we're going to go over exactly how the epigenome works, or how we think it works, in detail, in depth, all the different methylation, acetylation, ubiquitization, sumolation processes, right? We're going to talk about sirtuin genes, telomerase, telomeres, mTOR, AMPK. We'll go more into the folic acid and stuff. In the supplement and nutrition class, we're going to talk about different vitamin supplements. So we're going to address all of the vitamin deficiencies that we mentioned here are going to be addressed in depth in the nutrition and supplementation class. In the genome and epigenome class, we're going to go over in depth what methylation is, what acetylation is, how that stuff works, basically why eating good or eating bad affects us positively or negatively, right? And then we'll get into peptide therapy. Peptide therapy has been around for 30 years, you guys. These are natural peptides, or small proteins that are in our body that basically have 0 serious adverse health risks. And there is a huge amount of literature showing peptide therapy improves healing by a huge percentage. It can reduce or completely resolve things like MS, multiple sclerosis, Alzheimer's. There's a huge amount of potential by using these natural peptides in the body. The reason that nobody knows about these peptides is because the pharmaceutical companies can't patent natural molecules. They can't patent natural molecules. So if it's a natural molecule, the pharmaceutical companies can't make money off of it. If they can't make money off of it, it's either blacklisted or it's just not discussed at all, because they would rather make money than make people healthy, right? So what they have to do as far as changing natural peptides, so semaglutide, big area of interest right now, right? Semaglutide is a GLP-1 agonist, right? It changes our ghrelin hormone concentration being released, and it makes us less hungry, so we eat less food, right? The problem with Ozempic is it's not natural. It's like 50 times more potent than the actual normal semaglutide peptide, right? So Ozempic is 50 times more potent than the natural protein, right? So that's just one example of why people never heard about semaglutide, but they hear about Ozempic. It's because the pharmaceutical companies can't patent semaglutide because it's a natural protein. They need to change it a certain amount to be able to alter the structure to a point where they can get a patent for it. However, what altering the structure does is it makes it into a non-natural protein, which is why people have so many negative side effects from non-natural things that we put in our body. Natural peptides, in the peptide class, I go over all the literature, there's basically zero serious negative adverse events for any of the peptide therapies that are natural peptides, right? Death is not one of the possible outcomes. Death is listed 3 times in the side effects for ibuprofen. If you look at the full ibuprofen side effect list, death is listed 3 times. So these peptide therapies are more safe than ibuprofen, and they're a bazillion times more effective, and there's all sorts of ones that are used for different stuff. It's so cool.